Good morning, students. Today, let me explain you about a new topic from dairy chapter, that is collection, processing, and preservation of milk. And apart from these headings, it also includes marketing of milk. What is milk? Milk industry is an ancient mankind itself, as it is the substance created to feed the mammalian infants. It is considered as the most valuable and nutritious product for the human consumption. Most suitable and elaborate substance provided by nature. It is the best nutrient medium for the growth of various organisms. It contains proteins, fats, carbohydrates, inorganic salts, and vitamins. A brief composition of milk: water of 87%, proteins of 3.5%, wherein it includes mainly casein and whey. The fat content is about 3.5 to 3.7%, and the carbohydrate component is about 4.9%, which is chiefly represented by lactose. And it also has some vitamins like niacin, riboflavin, thiamine, vitamin B6, vitamin A, and D, that is depending upon fortification. Minerals like calcium, phosphate, magnesium, potassium, sodium, etc. And the enzymes includes lipases, proteases, and peroxidases. So this is a picture showing you how that milk is going to be transferred from the producer to the milk centers. So here you can see a person is pouring the milk that is collected by him into the big containers or the carriers, from which it is being taken to the dairy centers or the dairy industry. Once the milk is being received, it will be stored properly, and for that storage it has to be processed. So before going to those aspects, let us go through how milk is being collected. And when milk is brought from the farm to the dairy for processing, the following information on the milk is required. First one, quality, that is about acidity, pH, alcohol, and clot on boiling test. And the quantity, so how much quantity of milk is collected, that has to be recorded. And the composition, like total solids and specific gravity. And the presence of contaminants like neutralizers and preservatives, etc. And if there is any adulteration by means of fats, titratable acidity, and specific gravity test, etc. So after doing all these parameters, the milk has to be stored. Now let us go through what procedures are being followed. They are straining, filtration, and clarification. The object is to improve aesthetic quality of the milk by removing visible foreign matter which is unsightly and may therefore cause consumer complaints. Straining is separation of milk according to breed, health status of the animal, and so on. The filtration removes the suspended foreign particles by the straining process, and clarification removes the same by centrifugal sedimentation. So here you can see the picture wherein you observe the collection of a sample of the milk from different containers here, and that sample is being taken to the laboratory for testing it for various parameters. So you can see a person who is doing or subjecting the milk for various types of tests, and that milk which is being collected by this the containers, the big containers or the vehicles, wherein the ice is being added to it, to the storage tank, that is to preserve it until it is being shifted or transported to dairy industry. You see this transfer of milk from such storage vehicles to milk silo through pump. A brief uh, look at the milk processing operations. So once the raw milk is collected, it is being subjected for batch mixing, then to centrifugation and cream separation, then to heat treatment, that is pasteurization, and then to homogenization. And such a homogenized milk is being processed for two different steps. One is initially through filling, sterilization, and sterilized milk is obtained, and the other way is UHT treatment of the homogenized milk and aseptic filling, and that gives you the UHT milk. UHT refers to ultra high temperature. Milk is a sterile at secretion in the udder but is contaminated by bacteria even before it leaves the udder. Further, infection of the milk by microorganisms can take place during milking, handling, storage, and other pre-processing activities. Here you see some of the types of microorganisms that are generally found in the milk that we drink. One is Streptococcus lactylus, Achromobacter, Bacillus subtilis, and coliform bacteria. Coming to the preservation of milk, so in this slide you will be observing the section where the milk is being preserved, and the following methods of preservation are being followed. They are pasteurization, sterilization, and dehydration. Coming to pasteurization, the main purpose of pasteurization is inactivation of the bacterial pathogens, and the target organisms are Coxella burnetii. And pasteurization assures of longer shelf life for the collected milk, during which inactivation of the most spoilage organisms and of many enzymes. And pasteurization includes the heat treatment of 72 degrees Celsius or 161 Fahrenheit for 15 seconds, which is called HTST, or 63 degrees Celsius, that is for 145 Fahrenheit for 30 minutes or equivalent. It does not kill all vegetative bacterial cells or spores like Bacillus species and Clostridium species, and pasteurization temperature is continuously being recorded. What is pasteurization? The definition goes like this: it is a process of heating a food, which is usually a liquid or milk, to a specific temperature for a predefined length of time, and then immediately cooling it after it is removed from the heat. And this process slowly spoilage caused by microbial growth in the food. History of pasteurization. The French scientist Louis Pasteur invented pasteurization, and to remedy the frequent acidity of the local wines, he found out experimental that it is sufficient to heat a young wine to only about 50 to 60 degrees Celsius, that is to an extent of 122 to 140 degree Fahrenheit, and it is heated for a brief time to kill the microbes. Pasteurization was originally used as a way of preventing wine and beer from souring, and it would be many years before milk was pasteurized. Pasteurization of milk was suggested by Franz von Sochlet in 1886. Now coming to types of pasteurization. The first one, as I have mentioned already, HTST, that is high temperature and short time method. Second one, UTHT or UHT, ultra high temperature pasteurization. And the third type is VAT pasteurization. Coming to HTST, the HTST pasteurization standard was designed to achieve a 5 log reduction, killing 99.999% of the number of viable microorganisms in the milk. This method requires that the milk be held at 161 degrees for 16 seconds, and this process also referred to as continuous flow pasteurization and requires the milk to be processed or forced through metal pipes that are heated from the outside. Coming to UHT, it produces a product that has a stable shelf life of up to 2 months. UHT processing holds the milk at a temperature of 138 degrees Celsius to 150 degrees Celsius, and this is for a minimum of 2 seconds. Coming to VAT pasteurization is the most gentle type of pasteurization. Here the milk is held in a heated vat at 145 degrees Celsius for 30 minutes. It is then quickly cooled to 39 degrees. Next one is sterilization. Sterilization is a term referring to any process that eliminates or kills all forms of microbial life, including transmissible agents such as fungi, bacteria, viruses, spore forms, etc., present on a surface contained in a fluid. In medication or in a compound such as biological culture media, sterilization is generally being followed. Sterilization can be achieved by applying heat or chemicals or irradiation or high pressure and filtration or combinations thereof. Next one is Tyndallization. It is a new method and it is named after John Tyndall, which is a lengthy process designed to reduce the level of activity of sporulating bacteria that are left by a simple boiling water method. It involves boiling for a period of typically 20 minutes at atmospheric pressure, that is standard, and it is being cooled, incubating for a day, and boiling, cooling, incubating for a day. The three incubation periods are to allow heat resistant spores surviving the previous boiling period to germinate to form the heat sensitive vegetative stage, which can be killed by the next boiling step. This is effective because many spores are stimulated to grow by the heat shock, and the procedure only works for media that can support bacterial growth. It will not sterilize plain water. The next method of separation or the purification is membrane filtration, which is also called microfiltration. And as you see in the picture, microfiltration is a process of filtration with a micrometer sized filter. And it is a membrane technical filtration process which removes contaminants from a fluid by passage through a microsporous membrane. A typical microfiltration membrane pore having a size range of 0.1 to 10 micrometers. So this is the picture wherein you can see the microfiltration of the collected milk. When milk arrives at the collection center, information on the milk is needed, and this information could be quantity, quality, hygiene, composition, whether water has been added, etc. And this is needed to determine the amount of money that milk producer will get. The level of information required depends on many factors. Coming to milk sampling, it is an expensive to analyze all milk supplies on a daily basis for a parameters. To avoid this, you can sample the milk and place it in sample bottles with preservatives added, and potassium dichromate can be added to keep the samples fit for testing in a good condition. Testing can be done on a mix of these samples. Alternatively, milk can be sampled and tested on a random basis. Always try to keep milk samples cool, close to freezing point, but make sure that the milk is heated to 40 degrees Celsius and then it is cooled to 20 degrees Celsius and mixed before testing, and milk must be thoroughly mixed before sampling to make sure that the fat is dispersed throughout the container. Make sure that the samples are labeled and carefully recorded to avoid confusion. Now coming to milk testing. The methods used for milk testing are usually related to the payment system adopted. The different methods for milk testing are briefly described below, with an emphasis on simple and cost-effective models or methods, and these focus on milk reception tests that that can be carried out at collection centers. Quality testing. Milk testing for quality can be divided into testing for hygiene and for composition. You always have to balance between the costs and benefits of the test because testing regularly can become very expensive. Make sure you always clean milk testing equipment thoroughly after every usage. You can use boiling water for at least 1 minute, 70% alcohol, then keep the equipment in a flame, and some examples of tests in order to cost and simplicity are described below. First one, taste, smell, visual observation and temperature. This should always be the first screening of the milk since it is cheap, quick and does not require any equipment. And these tests are also called organoleptic tests. It is also reliable, and if the person carrying out the test it is experienced. The tester smells the milk, observes the appearance and tastes if necessary, and check the can for cleanliness, looks for sediment and filters and the milk to check its cleaning. If doubts arise after the examination about the quality of the milk, the other tests can be done to determine the quality. Second one, density meter or lactometer test. With a lactometer, the specific density of milk is removed, and at 15 degrees Celsius, the normal density of the milk ranges from 1.02 to 1.033 grams per ml, whereas water has the normal density of 1.0 grams. So when you read the lactometer, you can determine whether water has been to the milk or not. And it is best to combine the lactometer reading with the FAR test. Other tests can be done to determine the quality. That is density meter or lactometer test. When a lactometer, the specific gravity test milk is measured at 15 degrees Celsius. The normal density of the milk ranges from 1.028 to 1.033 gram per ml, whereas water has a density of 1.0 gram per ml. So when you read the lactometer, you can determine whether water has been added to the milk or not. And it is best to combine the lactometer reading with the fat test. If the results of the fat test are low and the density is high, then the milk might have been skimmed or it must have been mixed or contaminated. If the results of the fat test are low and the density is low, then water might have been added to the milk. And you can use the lactometer reading together with what you have been added to the milk. You can use the lactometer reading together with the fat percentage to estimate the solids non-fat, that is SNF, content of the milk, that is milk payment system method, etc. Coming to the other important features, it always reads the temperature of the milk first. The lactometer reading varies according to the temperature. Make sure you adjust readings as indicated in table 3 below. Then the next test will be clot on boiling. The clot on boiling test is simple, quick and cheap. If the milk is sour or if the milk is abnormal, having colostrum or mastitis milk, and the milk will not pass this test. Place the test tubes with 5 ml of milk for up to 4 minutes in boiling water or in a flame. Examine the tubes and reject the milk if you can see the milk clotting. Please note, at high altitude, milk boils at a lower temperature itself, and this test is not very sensitive to slightly sour the milk, and an alternative is the alcohol test. Next is alcohol test. If the milk is sour or if the milk is abnormal, the milk will not pass the alcohol test. You carry out the test by mixing equal amount, that is 2 ml of milk and a 68% ethanol solution made by mixing 68 ml of 96% alcohol with 28 ml distilled water. Milk that contains more than 0.21 acid will coagulate when alcohol is added. Next is acidity test. This test measures the lactic acid in the milk. If the acidity is higher than 0.19%, then milk quality is poor and cannot be processed. If the acidity is lower than the normal, then the milk is of poor bacterial quality, or sodium hydroxide or bicarbonate might have been added to here. So this is an introduction about how marketing is done. Dairy farmers produce milk to sell it for a profit, and management helps to reduce the cost of production. So that is always obvious. Management decisions are influenced by price, supply and demand trends for milk and dairy products. Markets available for milk and pricing structure and regulation of milk marketing. So this is a brief introduction about marketing strategy. So this is the flow chart showing the different steps that are involved here in this dairy industry during preservation and processing and marketing. As we observe here, the first one is howling, receiving, storage. Processing is done after clarification. Processing is done after separation and after standardization and as after pasteurization and management uh, activity having homogenization. Then vitamin fortification, and such a fortified packed nutrients, that is milk, it will be there in the filling machines. From here storage is happening, delivery also happens, and markets and human refrigeration or home refrigeration. So these are the different aspects about this preservation, processing and collection of milk. So with this I have completed one more topic under dairy chapter. Thank you students.